Oh, welcome. So in today's video, we are going to look at the concept of prokaryotic genome. Introduction to prokaryotic genome. What is prokaryotic? And of course, ladies and gentlemen, let's start from the definition of the genome. When we say genome, it's a complete set of genetics information, or is a sum total of all the genetics information resides in a living organisms. So the total number of, or the summation of all the genetics material, that is the DNA that are found in a particular living organism that describe the genome of that particular organism. And very interestingly, genome is the language of life. It is the genome that make one species differ from one another. It is the genome that make us as human species to differ from one another. The way I am, the way you are, the way other individuals are, we are males and females, the way males and females are, they are different. And all that is as a result of our genome. So today, we are actually going to look at the prokaryotic genome. And don't forget, when we said prokaryotic, there are organisms that doesn't have nucleus and membrane-bound organelles, which includes bacteria. Most of the bacteria, they belong to prokaryotic organism. So now, trying to look at the genomes of prokaryotic organisms, they are typically composed of single chromosomal DNA and a circular DNA. So it means that the prokaryotic genome is made up of two very important genetics material, a single chromosomal DNA and a circular DNA. So that is why if you can see clearly, this is the structure of prokaryotic cell. And you can see that we have the bacterial DNA. So this is the chromosomal DNA. And then there is a circular DNA, which is the plasmids. And they are all located at the cytoplasm of the prokaryotic organism because they doesn't have nucleus and membrane boundary. So no nucleus, no mitochondria, no Golgi body. So that is why these genetics material, they are resides or they are found in the cytoplasm of living organisms. So then in addition now, these prokaryotic cells, as I said, they lack membrane-bound organelles. And this lack of the membrane-bound organelles is what influences their genomic organization. The genomic organization of human or eukaryotic genome and prokaryotic genome are different because we have membrane-bound organelles. So our DNA are present in the cytoplasm and mitochondria. So because we have this membrane-bound organelles, that is why the organization of the genomic material in prokaryotic cells are different from our own. It is the lack of this membrane-bound organelles that is what actually leads to the genomic, or that is what actually influences the genomic organization. So then in addition, understanding the prokaryotic genomes is crucial for insight into evolution and biotechnology. You see, in biotechnology, it's a technology that's trying to use living organisms or any part of living organisms to produce products that are beneficial to humans. And of course, evolution is a process that involves the change of a particular species of living organisms over a long period of time. So understanding the prokaryotic genomes have actually give insights on how the evolutionary process takes place in living organisms and also biotechnology, because most of the biotechnological products, there are products that are produced using prokaryotic cell machinery. So the machinery, the genetics material, prokaryotic organisms, and the entire prokaryotic organisms are utilized in biotechnology for producing industrial products. And then now the next thing is the structure of prokaryotic DNA. Prokaryotic DNA is usually compacted with the help of proteins. So there are proteins which we call them nucleoid associated proteins. So they are the proteins that help to compact and organize prokaryotic DNA. Let's see it actually from here. You can see clearly here, this is the chromosomal DNA. The chromosomal DNA is a circular DNA, and it is double-stranded pieces of DNA, which is not actually surrounded by a nuclear membrane because it doesn't have nuclear membrane. And you can see clearly this is a nucleoid. So the nucleoid is the associated protein that compact or that houses the prokaryotic DNA. And in addition, we also have plasmids, and you can see them clearly also in the cytoplasm. The plasmids are small circular, and they are independent double-stranded DNA molecules. These plasmids can frequently be transmitted from one bacterium to another. In fact, because of these applications of this plasmid DNA, its ability to transfer from one bacteria to another bacteria, that is why it's usually used in the recombinant DNA technology. So it has this potential. It is double-stranded circular DNA, and they are very small, and they're independent of our double-stranded. They are independent. They're independent of double-stranded DNA molecules. It means that they are not actually linked with our or with the nucleoids with the chromosomal DNA in the bacteria. They have two different focuses. They have different focuses. So that is why they are small circular and independent double-stranded DNA. So they are independent of the double-stranded DNA that is actually associated with the nucleoid-associated proteins. And they have the ability to be transmitted from one bacteria to another. That is why it's exploited in biotechnology, recombinant DNA technology, and genetics engineering. And one very interesting thing in most of the organisms that we have, they are known to have introns and exons. So the introns is a part of the DNA that is not responsible for coding any protein. So they are the non-coding region. So when we said non-coding regions, it means that there are regions that they are not or they cannot be utilized to generate protein from those sequences. So prokaryotic DNA lacks introns, so that doesn't have introns. So the absence of these introns in most prokaryotic genes contribute to streamline the genomic structure of the prokaryotic cell. Then in addition, as we said, plasmid, that is small circular DNA fragment, can also be present. And this circular DNA fragment, they carry also additional genetics information. So there are some of the genetics information that are coming from the plasmid DNA. So let's look at the structure here. So you see, this is a typical example of prokaryotic circular chromosome. So it's not as like our own. And this is our own eukaryotic linear chromosome. So they are different things. So you see, there are also genes. They are ORIC, that is the origin of the replication. So this is just to tell you typically that the nature of our DNA and the nature of prokaryotic DNA are not the same. We have circular chromosomal DNA, while our own is linear chromosomal DNA. So then, genome size and complexity. So actually, each and every living organisms have their own genome size and complexity. Like human genome is the most complex genome. Let's look at it closely. So the prokaryotic genomes vary significantly in size because within the prokaryotic organisms, we have different species of bacteria. So that is why the size also varies significantly, and the size range from 0.5 to 10 million base pair. But if you take like human genome size, it's about 3 billion base pairs. It's about 3 billion base pairs. So look at it. This is a typical examples of Escherichia coli BA01 strain. And these are the genes that are found. All these are genes. Look at it, they are this CDS, it means coding sequence. So these are the genes. So these genes is about actually 5,000 mega base pair. That is the size of this Escherichia coli genome. So this to tell you that because they have circular, the chromosomes is circular or their genome is circular. So this is actually a typical example of the genome of Escherichia coli, which is one of the well-known prokaryotic bacteria. So this is the difference as genome. In fact, the number of genes that are found, especially like in this Escherichia coli, they have about 5,000 different genes. And in humans, we have close to 25,000 genes. Close to 25,000 genes. And the genome size can be as low as or as fewer as 400 genes. So the number of genes can also differ widely, with some bacteria having as few as 400 genes. So you see, different organisms have different genome size. Have different genome size. So you see, in this case, they can have as few as 400 genes. But very importantly, you should understand that despite the fact that they have smaller size, the prokaryotic genome can encode complex pathways and carry out various functions that actually sustain the life processes of that particular prokaryotic organism. So they have smaller genome size between 0.10 million base pairs. And most of them, those that have larger genome size, you see that most of the genes is around 5,000 genes maximally. But you can have some, as I mentioned, as fewer as 400. And that is enough to sustain the life of the organism. So despite their small size, the prokaryotic genomes, as I said, it can encode the complex pathways and carry out all the life processes. So then the gene organization and regulation. So the genes in prokaryotic genomes are often organized into introns, allowing coordinated expression. So you see what actually happened in the case of prokaryotic organism. their genes are coordinated together. They are always in blocks. They are in blocks. And the genes are expressed at the same time. Different genes, but exist in blocks, expressing different proteins in order to make sure that they carry out a specific types of function. Like, for example, we have a lac operon. A lac operon is an operon system where there are three different genes that express together, regulated together, in order to make sure that there is a proper lactose metabolism in the prokaryotic organisms. And very importantly, these genes or this prokaryotic DNA, it contains promoter and a terminators. So when you say promoters are the sites of the DNA where RNA polymerase is a major enzyme. RNA polymerase is a major enzyme that transcribe DNA into RNA. So these are the enzymes that convert DNA into messenger RNA. So that is a major enzyme that do this work. So where this enzyme first recognize and initiate transcription is the promoter region. So promoter region is the region where the RNA polymerase recognize, binds to initiate the transcription process. So the process where DNA is converted into mRNA is called transcription. And the master enzyme that carry out that function is RNA polymerase. And this RNA polymerase binds to promoter sites. And then the terminator site is a site where the RNA polymerase will be signals to terminate the transcription process. That is to stop the transcription. So these two regulatory elements are very important and play a critical role in gene expression control. Because it is with these two regulatory elements that your DNA will initiate transcription, that your DNA transcription will be stopped. And that is why for every DNA, we have start codons where the transcription starts. So they start codons because we have what we call genetic codes. And it is those genetic codes that are also serve as a major signals in initiation or in initiating transcription, or that start as a transcription point. So the base or the initiation codons is AUG. That is AUG, which actually code for methionine as an amino acid, methionine. It is the starting amino acids during protein synthesis. And then, of course, there are terminators where we have stop codons. But more importantly, what I wanted to say here, this is usually happens during protein translation. So for the terminators, we have two types of terminators. We have two ways where the RNA synthesis will be terminated. We have row-dependent termination and we have row-independent termination. Row-dependent and row-independent termination. So as you move to the higher level in 300 level, both the student or all the students in life sciences will know more about this, because this is just to give you an idea of how the structure of the prokaryotic gene are. So we have row-dependent and row-independent termination. So this termination is just to send signals to the RNA polymerase to stop transcription. So more importantly here, this organization facilitates rapid adaptation to environmental changes and availability. Because one very interesting thing that we should understand, when we said that operon, so all the genes are expressed together in order to make sure that a particular system or a particular functions is maintained. So they are all expressed together. So like, for example, now, let's look at this example. You see clearly here, there are genes A, B, C, and D. This is genes A, B, C, and D. You see, so they are all in blocks. So they all express together. First, they transcribe together to produce what we call polycistronic messenger RNA. So produce when we say cistronic, it means RNA. So polycistronic messenger RNA. Because here you can see clearly, why is it that it is called polycistronic messenger RNA? You can see clearly that there are 4 genes here. So it means that 4 mRNA will be generated. So that is why it is called polycistronic messenger RNA. So that is why the polycistronic messenger RNA are only produced in prokaryotic, or is only present in prokaryotic organism, not in eukaryotic, because all the genes are in blocks. They express together, they transcribe together, and they translate together to produce the proteins, which subsequently all the proteins will be involved in trying to make sure that one particular metabolic pathways is concluded. You can actually go and watch my video on a lac operon, that is lactose operon, where 3 genes are expressed, and those genes are all trying to make sure that there is a proper transport and the metabolism of the lactose. So all the genes are expressed together, they are transcribed and translate together to produce different set of proteins, but with the purpose or with the same purpose. And this is the regulatory gene. That is the site where the genes can be regulated. So all these genes, A, B, C, and D, can be regulated together. And we look at it, these are the structural genes. So why they are called structural genes, it means that they are genes that are trying to maintain the structure of something, maybe a cell or something in the body or in the prokaryotic organism. So this is the regulatory gene. So a regulatory gene is a gene or is a region where all these genes can be regulated, because they have one promoter and they have one regulatory site. But unlike in eukaryotic organism, each gene have its own regulatory point. Each gene has its own promoter site. But in this case, in the case of lac operon, or in the case of operons, and operons is a DNA, or it's a gene, or it's a prokaryotic genomes where more than 1 genes are expressed together under the same promoter using the same regulatory region. So that is why in operons, there is what you call a repressors. We have activators, we have activators, we have repressors, we have activators, and then we have inducers. Inducers. So what are the functions of each of these? The repressors, there are proteins family. There are proteins family, you should get it. There are proteins family that bind to regulatory region and suppress transcription. Suppress or inhibit transcription. That is what we call repressors. While activators, there are proteins molecules that increases or activates transcription process. While inducers, there are molecules that can either activate or inactivate transcription process. So ladies and gentlemen, we should understand the operand system. There are genes that are expressed, there are more than one genes expressed together with the same purpose under one promoter using one regulatory region. So that's why you can see you have gene A, gene B, gene C, gene D, and then we have transcription, which leads to generation of polycystic messenger RNA. It is poly because it is messenger RNA of different proteins that conjugated together. And then those conjugated messenger RNA express differently to produce 4 different proteins for the same purpose. So this is actually brief on the concept of operand system. So then the next thing, let me just actually show you another examples. This is prokaryotic and eukaryotic, and you can see clearly there is one promoter region where we have enhancer and we have repressor. So activator can be called enhancer, and then this is the repressor. So this is the regulatory gene. So regulatory gene on its own, regulatory gene on its own has its own promoter. It has its own promoter, and the activation of this promoter will activate the regulatory gene, which subsequently the regulatory gene, depending on the regulation, if it is enhancer that bind to the promoter, it means that the regulatory genes will be activated to promote transcription. If it is repressor, there are molecules that will bind to the promoter and activates this regulatory gene in such a way that the operand or the transcription of the genes will be blocked, will be suppressed, or will be decreased. So you see, this is how we have, you see, we have different genes, gene A, gene B, gene C, gene D, gene E. All of them, you see, there are 1, 2, 3, 4, 5. So these are 5 different genes, and they are going to express together. And you can see clearly there is no any introns, as I mentioned. It doesn't have introns. All of them have exons. But look at the eukaryotic gene structure. So in the case of eukaryotic gene structure, you can see clearly that there is enhancer, there is a terminator, there is even silencer, enhancer, silencer, and there are genes. And it has both introns and exons. So exons is a coding region of the gene, while introns is a non-coding. So that is why usually there is a splicing. And there is also a poly-A tail sequence. A poly-A tail sequence is a sequence that aids in stabilization of RNA. So it aids in stabilization of RNA. So the most important thing for the for this structure is that you should understand that in the case of eukaryotic gene, the genes are not in blocks and they are not expressed together, and each gene has its own promoter and they are not expressed at the same time. So that's the most important thing that you should understand. So then the next thing is the promoter that we are talking about. So the promoter that we are talking about, the promoter sequence is present in the upstream region of the gene, that is near 5' end. So remember, whenever we have a genes or the DNA is started from 5' to 3', and then we have 3' also to 5' direction. So in the promoter region, there are the upstream region. Let's say this is the DNA. So you may have here as your promoter. So you call it upstream region. And here you can call it terminator, which is the downstream terminator, which is known as the upstream, the downstream region. So this promoter region is made up of -3' and -10' So the -3' is made up of TTGACA, while -10 is called TATAAT, that is TATAAT. They are known as a promoter sequences that initiates the process of transcription by interaction with the RNA polymerase. So this -5 and -10 region or sequences, they are the sequences that interact with the RNA polymerase, initiates transcription process. So you see the -5 and the -10 region, we call them consensus sequence. We call them consensus sequence, which means they are conserved sequence in man organism. So you see it is consensus sequence and it is conserved, that it is present in all human. And they are the region that are recognized by RNA polymerase to initiate the transcription process. So the -10 region is known as the Rivno box. It is known as Rivno box. So this is actually how we have it. So this is the recognition sites. That is the -5, -35, and -10 region, where the RNA polymerase binds to initiate transcription of the DNA into RNA. So ladies and gentlemen, this is actually a description of prokaryotic genome, and I hope this video will be very important to all of you. And please, if this is the first time you come to my YouTube channel, please try to subscribe and join the YouTube community. So bye bye and see you.